Good morning, everyone. I'm very happy to introduce Dr. Mark Lever as our speaker today. Mark is an associate professor at the Marine Science Institute of UT Austin, and he is an adjunct professor at Center for Geomicrobiology at Aarhus University, Denmark. Before joined UT in 2022, he was an assistant professor at UTIG, Zurich. And he got his PhD in marine science from University of North Carolina at Chapel Hill in 2008. And Mark is both a microbiologist and a biogeochemist. And he is interested in the role of microorganisms in Earth's carbon cycle. With that, let's welcome Mark to start.
Thank you very much for inviting me and for the great welcome and all the wonderful meetings I've had here so far. So I very much appreciate it. And thanks for organizing my visit, Kehua. Today I'll be talking about work that's been close to my heart since I was a graduate student. As you can see up there, that was me in actually 2004 on my first ocean drilling expedition, the JOIDES Resolution. And I'll be talking about a project that's kind of been ongoing. It was a focus of my PhD, and then I've kept working on it in the background as I've gone into other fields since then over the past few decades. So the topic is population dynamics of methane-cycling microorganisms in subseafloor sediments. So before I go into that, I'll give a little bit of background of the kind of work that my lab does. So the key question that drives all of our research is what determines the fate of organic carbon in the biosphere. We do work on coastal sediments, in Port Aransas on the Texas coast. I've done quite a bit of work on ocean drilling expeditions further offshore. While in Switzerland, I started looking at anthropogenic impacts on the carbon cycle in lakes. I've done some work on Earth's crustal environments and also mantle-influenced environments on Earth. And more recently, I've been looking at, or I'm starting to look at the methane cycle in coastal macrophyte environments and also doing more applied work related to the farming of seaweed on the Texas coast. But ultimately, to me, these may seem like very different subjects, but ultimately, this is what comes down to that I see as being the central question. What determines the fate of organic carbon in the seafloor? And overall, most of our work is on sedimentary environments. And we want to understand what happens with organic matter that settles and sinks to the bottom of the ocean. What determines whether that organic matter goes to CO2 or methane? And that's because of microbial activity that break down and mineralized organic matter. What determines whether it becomes buried in the long term, contributing to this vast carbon sink. In order to address this question, which is very interdisciplinary, we study what microorganisms are there. And so focusing on bacteria and archaea that are involved in the degradation of organic carbon, we look at the rates at which organic carbon is being degraded using isotopic approaches, looking at specific genes that are indicative of specific carbon degrading reactions. We also measure rates of activity using simple geochemical and at times enzymatic assays. And we combine this with a geochemical analysis of organic matter compositions and electron donor and acceptor concentrations. And all of this combined is what is our approach to understand what determines with organic matter in the seafloor. And we have a strong quantitative component, so we're always trying to measure rates and inventories of the bacteria, of the organic compounds when we're performing these analyses. And a longstanding interest, because of my work in ocean drilling, has always been also what's the impact of the underlying oceanic crust and also mantle environments on processes that are happening deep in the sediments. So a little bit of background on how does microbial breakdown of organic matter work. So we always, in many environments on Earth, most of the seafloor, the main energy source for microorganisms is biomass produced by photosynthesis, and that then undergoes various transformations through the marine food webs. Some of that a small portion in most places of that organic matter makes it down to the sediments, where it's present as macromolecules such as polymers, carbohydrates, proteins, DNA, RNA, lipids. And that's where then the breakdown of organic matter really starts, at least that's where it gets interesting, in my opinion. The first step that is always happening is we need extracellular enzymes that are released by certain groups of bacteria and archaea. They release these enzymes out of the cell, and they act like scissors that chop up the polymers into smaller components, such as monomers, that can be taken up by the cells. That uptake is essential for the organisms to gain energy. They can only gain energy from what they eat and ingest. And we have very different scenarios then. So once that organic matter is imported into the cell, when we have oxygen present, we can have a more simplistic food web or food chain where we have a one-step aerobic respiration to CO2. When oxygen is absent, which are the environments that I mostly study, we're getting a much more complicated picture here. We have specific group of organisms called primary fermenters that are specializing in the initial breakdown of monomers to alcohols and organic acids. We have secondary fermenters that then take those alcohols and organic acids and produce C1 compounds such as methanol and methyl sulfides, formate, and hydrogen. And then the last step is what we call the anaerobic respiration, which is the final step that produces that CO2 and methane and converts the last bits of organic matter to CO2 and methane. And today I'll be talking about those organisms that are involved in the microbial production of methane. And as far as we know, these are all archaea, so they're distinct from bacteria. They're in their own fundamentally different group of microorganisms. And when we look at what are the archaea eating, it's the bits and leftovers of fermentation, and the most important ones are hydrogen and acetate, at least according to the literature. Now, where do these archaea, where are they active? Well, as it turns out, that methanogenesis is a very low energy reaction that is typically confined to deeper layers, where all the other anaerobic respirations, such as denitrification and manganese reduction, iron reduction, and sulfate reduction, where all of those processes have run out of their main electron acceptors. And only then, because methanogenesis is such an energy-poor process, does it become the dominant respiration reaction. So it's typically sediments where sulfate has been depleted. Importantly also, while methane production in marine sediments could be a huge source of the greenhouse gas methane to the atmosphere, we're lucky that almost all that methane that's produced down there is actually consumed deep within the sediments by relatives of methane-producing archaea, which call them anaerobic methane oxidizing archaea. And they work together with sulfate-producing bacteria to consume all that methane in what is known as the sulfate-methane transition, which is right where methane concentrations meet sulfate from overlying seawater. Important also is that nothing is ever as simple as it seems. There are also some methanogens that are living up here, but they are minority populations that are eating less common carbon sources, such as methanol and methyl sulfides, that the sulfate reducers, for the most part, do not eat. So that's just a little bit of background here. Now, even though methanogenesis is a process that's restricted to these deep layers where all the other processes have run out of electron acceptors, it has a huge role in Earth's climate. And subseafloor sediments harbor Earth's biggest methane reservoir. A lot of that is in the form of hydrates. And eruptions of these hydrates have been linked to past climatic shifts on Earth. So even though this is a process that's really restricted to the deepest layers of sediments where there's almost nothing to eat over geologic timescales, the amount of methane that's produced is huge. And when that methane has erupted, it's had major impacts on the Earth. So the methane hydrates that are then formed when you have high methane accumulations are mainly along continental shelf and slope regions, where we have a combination of low in situ temperatures, close to zero, and we have high hydrostatic pressure, and that combination results in the formation of these hydrates. And that's where most of the methane ends up being stored over geologic timescales in subseafloor sediments. The importance of this is manifold. So there have been a number of studies, and I'm not an expert to know how much of this is is accepted or speculation, but there have been a number of studies that suggested that past changes in sea levels that have triggered eruptions and melting of methane gas hydrates and bubbling of methane to the atmosphere has, for instance, terminated ice ages. And many other examples exist where these methane hydrates are believed to occasionally have played a major role in controlling Earth's climate. Now, my work is more on not looking at climate, but looking at who's producing this methane. And it's important to know that even though these organisms are so important, almost all the research on these organisms so far, if you go into the literature, has been on very specific types of environments that don't represent most of the seafloor. And those are hydrocarbon seeps, methane seeps, cold seeps, hydrothermal sediments. And also there's been a considerable focus in some places on just on the process of anaerobic methane oxidation in shallow sediments. What is striking and weird is that with all the ocean drilling expeditions that have happened, that have had a strong microbiological focus over the past decades, there's been very few findings, detections of methanogens in these subseafloor sediments. And I think that's really remarkable, considering that this is the dominant respiration process in these deep sediments. And one thing that's notable is that most of these assays that were used were targeting all the DNA in the sediments, so using metagenomics, which is a process where you're sequencing all the DNA that's in the sediments, or targeting 16S ribosomal RNA genes, which are present in all bacteria and archaea. So taking this universal approach to see whether methanogens are present and who's there. And I think perhaps, or based on my data, that is perhaps part of the problem, why they haven't been detected. So the hypotheses I'll go into today are that methane-cycling archaea are rare, but very important members of the deep biosphere and commonly missed by these general surveys that target everything. We need assays that specifically target these organisms to be able to detect them. The next ones are that these methane-cycling archaea, they follow clearly the sulfate gradients in the subsurface sediments, and that other factors that we would expect to be important, such as in situ temperature, lithology, and organic matter sources are also important drivers of the community structure of these archaea. And lastly, that we can actually get growth of methane-cycling archaea deep, deep below the seafloor. Once the sulfate is gone and the conditions are right, we have this geochemical change that's driven by geologic processes. And that may sound like to geologists like a no-brainer, but to microbiologists, there's been a long discussion whether there can be any growth in subseafloor sediments. So many microbiologists have argued that subseafloor sediments are basically a death curve, and over time there's no growth. You just have survivors and fewer survivors over time. And so we'll see what we found. So the sampling that we've done, and at least the first two studies I'll be presenting was done with the JOIDES resolution, and starting with Leg 201, which is the very first drilling expedition ever to have a strong microbiology focus. And this was a cruise that took place in 2002, and where the main target was the Peru margin and Peru and Equatorial Pacific. And I'll be talking about a site in the Peru Trench, ODP site 1230. This site is remarkable in that it has a water depth of 6 kilometers, but nonetheless has very high rates of microbial respiration for a deep sea sediment. So if you look at the dissolved inorganic carbon concentrations, they reach over 180, maybe even 200 millimolar, which is incredibly high. Sulfate concentrations are gone at 8 meters below the seafloor. And then below that, we have rapid accumulation of methane, and then we even have methane hydrates in deep subseafloor sediments at this location. So here just shows you those crucial zones of where we would expect the sulfate-dependent anaerobic methane oxidation in red, and I'll be using these colors in the later slides also, and the zone of methanogenesis below that. So several studies had tried to see if there are methanogens there using general assays, but failed. And this raised many questions. One was, maybe methanogens have cell walls that are really, really rigid and strong that we cannot crack with our DNA extraction methods. So another one is, maybe these are not the typical methanogens we know. Maybe it's some bacteria that have acquired the ability to produce methane and are responsible. And the third one, which I'll be focusing on, which just to give away the results, is that methanogens just happen to be incredibly rare in these environments. And to address that hypothesis, it was key not to look at all of the DNA, but specifically at a gene that is diagnostic of methane-producing and methane-oxidizing archaea. It's the gene that encodes methyl coenzyme M reductase. And the idea was by targeting a gene that's really specific to these groups, we can detect them even if they only make up 1,000th or 10,000th of the total microbial community. And this is a really good gene because many other genes are passed along between unrelated organisms, so you can really not say which organism it was when you sequence it. With this gene, it appears to have only been vertically inherited. So when we look at a phylogenetic tree, that's a universal phylogenetic tree based on 16S ribosomal RNA, it matches that of MCR8, suggesting that we can really use it to identify the organisms. So now getting into the subject matter, so we were able to detect with a lot of optimizations, we were able to detect a very low diversity of only 3 different groups of methane-cycling archaea. The first group is methanothrix, which are known as methane producers that use acetate. They split acetate into CO2 and methane and use this to conserve energy. And then we found 2 other groups, the NME1AB group, which is known to be a methane-oxidizing bacterial archaeal group, and then a new group related to NME1AB that is one generally has assumed to be methane-oxidizing, but we don't think that's what's happening. So without further introduction, here's the distribution that we found of these groups relative to the presence of sulfate, the sulfate-methane transition, where methane oxidation is happening in the methanogenesis zone. So what was striking is that we don't find any of these archaea in the sulfate zone, but once we reach the sulfate-methane transition where we have anaerobic methane oxidation, the NME1AB group popped up, which is kind of where you would expect to be, right? Where methane oxidation is happening, that's where methane oxidizing archaea are showing up. Below that, we started seeing in deeper layers the other groups, the methanothrix, the known methanogens, and the NME1D group, which we don't really know what it's doing. And we're able to find these groups all the way through the remaining sediment columns. So here, these are the different depths that we tried. And in most depths, we were able to detect them. And we're able to show that indeed these organisms, the potential producers of methane can be detected with these very sensitive methods, and they are there. Now, looking at these sequences and based on known physiologies of these organisms, there's still some things that are unclear. So, and kind of contradictory. So the first one is that when you measure the delta 13C of dissolved inorganic carbon in these sediments, it indicates that not acetate is the main source, but CO2 reduction with hydrogen. And that is contradictory because the most abundant group, the methanothrix, are using acetate. Now, I've dealt with, this was a major thing that was holding me back from publishing it for a while, but in recent years, it's actually been shown that methanothrix, under certain conditions in the lab, can switch to CO2 reduction, not using hydrogen, but using direct electron transfer. So basically using electricity to reduce CO2. So maybe that explains that. With the NME1D, which we would historically thought to be methane oxidizers, it also turns out that they seem to be mostly in methanogenic sediments. So maybe they actually are methanogens. So that's where we're at with that. But there's, of course, many unknowns. We would actually have to do experiments and genomic analysis to really confirm this interpretation, which is based only on one gene, which doesn't really tell you much about the physiology of the organisms. So with that, our data then points towards methane-cycling archaea being very important, but very rare members of the subseafloor, and that they clearly follow these sulfate gradients, kind of what we would have expected based on other environments where they also seem to be restricted to deeper layers where sulfate is absent. The next thing we want to investigate was, are there other factors apart from sulfate that are driving the community structure, and looking at a very different environment that was a perfect case study, I think, to look at how temperature, lithology, and organic matter sources affect methane cycling. And the location here is the Juan de Fuca ridge flank off the coast of Washington State and Vancouver Island. It's a site named Site 1301. It's on the ridge flank, about 100 kilometers from the spreading center, from the Endeavour axis. It's a sedimented ocean crustal environment where we have 265 meters of sediment overlying the ocean crust. And it's a really interesting environment because we have a very strong influence of the underlying ocean crust on the deeper layers of the sediments. So this is a place where we not only have sulfate coming in from the overlying seawater and diffusing into the sediments where it gets consumed by microbial activity, but we also have sulfate diffusing from the underlying basalt, from the ocean crust into the sediments, where it then also gets consumed. And as a result of that, we have this kind of interesting sandwich profile where we have two sulfate reduction zones, one at the top and one at the bottom, and methane accumulation only taking place in between them where the sulfate is gone. And in this case, we try to quantify using an older method the copy numbers of MCRA genes, and we found some interesting trends. Well, we did find them throughout the sediment column, but with peaks not where we expect them. So the highest numbers were actually in the sulfate zone. I will not go into this any further today, however. So that's something for future research. So the methanogenesis zone was, again, confined to this interval in the middle of the sediment column. We then have these two methane oxidation zones, and then the sulfate reduction zones at the top and the bottom. And before I talk about the organisms that we found in the sediments, I'll go back one step, and that is back in 2013, we published a paper in Science where we documented the first detection of methane-cycling archaea in basaltic oceanic crust from this location, where we looked at specifically fissures and fractures in the basalt where there's water flow, and found that there are two groups of methane-cycling archaea present. Those are, shown in magenta at the top here, a group named Methanoperidinaceae and a group named ANRI1AB. Back then, we concluded that this group is a methanogen, this group is a methane oxidizer, and that we have a closed methane cycle. Since then, we know, based on culture evidence, that Methanoperidinaceae are actually methane oxidizers. So it's not correct, probably what we wrote in the paper, which raises the question, where did the methane come from? Because perhaps we have two groups of methane oxidizers, but no methane producers. It raises the question, is the methane coming out of the rocks produced by some kind of, we're talking 64 degrees Celsius, low temperature processes, or is it outgassing? Or another question, possibility is that is actually the other way around, that ANRI1AB are the producers of methane. We don't really know. However, in the overlying sediments, interestingly, we found the same groups shown at the top here. So in terms of total numbers of sequences, the same groups and very close relatives of the ones that were in the basalt were found throughout the entire sediment column. And in addition to that, we found a much higher diversity. So shown here in magenta are all the other groups that we detected. So this raises, of course, the question, how connected are these two environments? And also, a basaltic environment is completely different in many ways from a sedimentary environment, and why are we finding the same methane-cycling archaea there? But I'll get into that more. So here are the results, and I know there's a lot to digest, but if you just look at the yellow bars and the blue bars, those are the two groups we found in the basalt. And when we go down from the sediment surface to the deepest layers of sediments, these are the dominant groups of methane-cycling archaea we found. We had to use two different assays to target them. And interestingly, those other groups that were detected, so we found methanothrix again, we found other methane oxidizing archaea, ANMI2 and ANMI3. Those were all confined to the methanogenesis zone, or the lower methane oxidation zone. One thing that's striking is when we look at the in situ temperatures, they go from 2 degrees Celsius at the sediment surface to 64 degrees Celsius, and still it's the same groups. We're expecting probably there'll be some thermophilic methane producers showing up in a complete change in the community. And that would match what is traditionally known about microbial temperature ranges. So in the classic microbiological literature, we split microorganisms into being psychophiles with temperatures of 0 to 20 degrees as their growth temperature, 20 to 40s being mesophiles, 40 to 60 or higher thermophiles. That's not what we're seeing here. It seems like these organisms can grow at all of those temperatures. And there's, to my knowledge, not a single microbial isolate in the lab that's ever been shown to grow over a temperature range of 60 degrees Celsius. But here they seem to be there, and we do see methane production, so raises suggest they are active, and they're somehow able to live across this very wide temperature range. Interestingly also is, here I'm just showing the grain size data, but we have turbidites here, and these coarse-grained layers that are in dark, that are dominated by highly degraded terrestrial organic matter from the North Cascade Mountains, which are nearby. It's almost pure terrestrial organic matter, and then we have layers that are what I'm calling clay here. Those are dominated by marine organic matter. It doesn't seem to matter for the methanogens or the methane-cycling archaea what the source of the organic matter is. What really seems to matter is whether sulfate is present, but even that, we find these two groups even throughout the sulfate reduction zone. So it seems like we have two hypergeneralists that can just tolerate a very wide range of conditions. So surprisingly, we do not see any influence of temperature or lithology or organic matter sources on the community structure of these organisms. That leads me to the last question or hypothesis, which is that growth of methane-cycling archaea occurs at geochemical transitions centuries or longer after their initial burial at the seafloor. And I'll be now going away from ocean drilling to continental shelf environments off the coast of Denmark. So sites, 4 different sites that are in the Norwegian Trench, on the edge of that trench, and then going into the Kattegat region and the Lillebælt region off of the Danish coast. And the 4 sites I'm going to be presenting, sorry, this is not very, I'm going to just walk you through this. So these 4 sites differ tremendously in terms of the organic matter content. So if we will, this is our AU1 is our oligotrophic site with very little organic matter in the sediments. AU4 is the highly eutrophic area with tons of organic carbon inputs from seagrass and marshes and phytoplankton. So we have this gradient in the organic matter content, which is shown in black here. We have this increase in organic carbon in the sediments, and we can also see microbial activity increases corresponding to the organic carbon content. So in red are dissolved inorganic carbon concentrations, and those increase with depth and from sites with low organic carbon to high organic carbon. So we have a very big change in overall rates of organic carbon degradation. And this is reflected in the pore water geochemistry of sulfate and methane. So at our oligotrophic site, we have no methanogenesis zone. This is sulfate throughout the entire sediments. And then going from AU2 to AU4, we have an increasing shallowing of the methanogenesis zone towards the seafloor as the sulfate gets consumed more rapidly because of the higher microbial activity. Now, we see very strong changes in microbial communities, methane-cycling archaeal communities across these sites. I'm not going to go into that, but these raise the question, what drives these changes in the communities? And it seems like whenever we reach sediments where sulfate is, where we either have methane oxidation, so the red zones, or where sulfate is depleted and methane production takes place, that's where we have big changes in the communities of archaea, methane-cycling archaea. You can just look at the colors here. So the blue color here indicates sediments where we either have methane oxidation or methanogenesis, and predominantly the red and green taxa, though not everywhere, at sites where sulfate is present. So there seems to be this general change. Also, the yellow ones only show up in sediments where no sulfate is present. So the question then is, when you see a change like that, could this be because you have just selective mortality, that certain groups are maybe present, the blue groups are always present in the surface, but they are surviving at a higher rate, and that the ones that are dominant in the surface are dying faster with depth? Or could it be that you actually have growth of these taxa in subsurface sediments? And to look at this, we analyzed using a method called quantitative PCR, looked at different groups of methane-cycling archaea. We found certain groups dominated in surface sediments, but those will disappear with depth, so their gene copy numbers decrease, suggesting they're dying. But then other groups actually showed increases by 3 or 4 orders of magnitude with depth. So that's mainly our NMEs, our methane oxidizing archaea. They, as we would expect, increase to the sulfate methane transition where the methane oxidation is taking place, and then show some slight decreases. They don't disappear, however, which is interesting. And then we have what we think are methanogens. And those show an increase in the deepest layer. So once the sulfate is gone, that's when they reach their highest abundances. And we're seeing 4 order of magnitude increases, which to us clearly suggests it's hard to argue that this is selective survival. There must have been growth of these taxa. And then we have some ubiquitous taxa that we find everywhere at low abundances, but don't seem to follow any kind of trends. So just to reiterate, we see clear signs of growth, and these are sediment layers that have been buried for several centuries of methanogens and anaerobic methane oxidizing archaea, suggesting the deep biosphere is not just a dying biosphere, but it's a dynamic biosphere with microbial growth, and that just changes in the geochemistry are the main drivers of these abundance changes over time. How long back in time can we go is a big question. Could be if we had sediments where the methanogenesis layer starts in layers that are 10 million years old, would you still see growth? We don't know. This is only a few centuries, maybe 800 years or so, which is a long time, but maybe not that long. So that's a question for future research. So, but just to sum up, we see that there's growth of methane-cycling archaea in subseafloor sediments centuries after burial. So this brings me to my summary. We observe that methane-cycling archaea are, despite being really important in the global carbon cycle, members of what we call the rare biosphere. They're less than 0.1% of the microbial community based on the gene copy numbers. So but yet playing such an important role in driving, being the terminal remineralizers that are playing an essential role in consuming the acetate and hydrogen that's produced by fermentation. If we really want to study them, we could sequence just a lot more than we're doing right now, or we can use targeted assays. In this case, that's our preferred method, using a gene that's really unique to them. We can fish out and get rid of all the other stuff, only focusing on DNA of these organisms. It seems that sulfate concentrations and organic carbon contents are key drivers of the community structure of these organisms and their abundances. Interestingly, organic matter sources, lithology, and temperature seem to not play a big role, at least in these subseafloor environments. With organic matter sources and lithology, since we've done work that suggests that potential reasons, or we think the reason for this is that methanogens are not the primary degraders of the organic matter. So when you have terrestrial organic matter, it will have chemically very different compositions from phytoplankton-derived organic matter. But the methanogens are not eating that. They're eating the last breakdown products, the waste products, and those are hydrogen and acetate, methanol. They're universally produced. And so maybe for that reason, they don't care what the original organic matter was made. So most likely you see a change, and we do see it, differences in bacterial and archaeal communities among those groups that are involved in the first initial degradation step. So you have specialized groups that degrade certain types of terrestrial organic matter versus types of phytoplankton-derived organic matter. For lithology, may also just not matter because they're not using the grains, sediment grains as electron acceptors, or it's not serving any key purpose. We do see that methane-cycling archaeal communities respond to burial-related geochemical changes through mortality in some places and growth, and that's the growth part is what was the big finding here. And this also suggests that over geologic timescales, the subseafloor deep biosphere undergoes dynamic changes. These are very slow timescales, but they are there, and it's not the death curve that it's been proposed to be. Just a few slides on where we're going now. So this is previous research. We recently started looking at the distribution of methane production and methane-cycling archaeal communities and coastal sediments here on the Texas coast. It's a collaboration with Sean Gulick and John Goff from UTIG. And recently we had the inauguration of our new drilling vessel, the DV Manatee. That was Sean's name suggestion. And we're currently coring different places where chirp data indicate gas fields to be present in our local waters. So this is one of our next, or a big focus of our work related to methane cycling over the coming years. Look at what drives the communities of methane cycling archaea in coastal environments, and also a little bit of shameless promotion, we have the very first Southern Geobiology Symposium in Port Aransas this November. And this is, we particularly encourage graduate students and early career scientists to come, but everyone is very welcome. So we hope some of you will, or many of you will come to Port Aransas for that. All right, that's it for me.
And thanks, Mark, for teaching us on methane cycling. We have 24 minutes for questions.
Oh, sorry, that was fast.
Thank you. That was a great talk. I have multiple questions. The first question is, can you distinguish between like active biomass or biomass that's deceased or dormant if you're looking at the DNA? I mean, obviously the DNA will degrade eventually, but there must be some kind of window where this hasn't taken place yet. And the other question I have these kind of archaea be cultured with acetic acid in the laboratory, and what is their doubling rate and how much biomass do they produce? And have you done that personally?
Great question. So regarding DNA, yes, DNA can persist for long time scales in sediments, but the degradation rates of DNA are very temperature sensitive. So we have DNA hydrolysis and depurination reactions, which make it impossible to sequence or amplify the DNA. There's a very strong temperature dependency. So when we're looking at sediments that have 64 degrees Celsius that are over 1 million years old, we would not expect to detect any clean or amplifiable DNA any longer.
Yeah, I have like a follow-up question to that. So assuming the DNA degrades, and for example, I mean, could you find like residue, like purines, for example, that you could use to reconstruct how much DNA was there? I know that's maybe a very silly question, but
we can measure total DNA concentrations, and that would include also small bits and pieces, single purines and pyrimidines. We can do that, or short fragments of a few tens of base pairs or so that we cannot sequence. Certainly, we can do that. When we've done that in the past, they agree actually very well through quantitative PCR results. So with quantitative PCR, we're amplifying larger fragments that require fairly intact DNA. So they correlate very strongly with the copy numbers. So I think that, of course, you could argue that the temperature argument applies to Juan de Fuca ridge flag, where we have temperature of 60 degrees. And we can do calculations to show there really shouldn't hardly be any DNA left at 50 or 60 degrees Celsius after 1 million years. There shouldn't be anything left that is degradable. For the pure trench, you can say, well, that's not the case. It's 2 degrees Celsius there, or maybe 5 in the deepest layers. But I mean, we do have methane production, and we have methane hydrates, and we know they're biogenic. So that right there means clearly there's been biogenic production. Whether all the DNA that we're detecting belongs to active methanogens nobody can say for sure. Interestingly, at the Peru margin, I actually also did RNA extraction and was able to do reverse transcription of RNA into DNA. And I did I was able to detect messenger RNA at some of the depths. So messenger RNA is oftentimes what people will argue is a much more robust or sensitive indicator of whether an organism is alive and active, because it has a very short half-life. So within active microbial cells, messenger RNA has a half-life of a few minutes. Whether that's the case in the deep biosphere, we don't know. Perhaps not. That would not be very practical, because that's a very high metabolic rate that has to sustain that. But we did that. Compared to controls, we found clearly elevated copies of messenger RNA compared to any of our controls. And so I think that's another piece of evidence that they are active. Does that answer your first question?
Yes, that answers my first question. My second question was just about culturing, but we have a meeting later and we can talk about culturing.
So the culturing, what's interesting is that methanothrics we find there are not, they have fairly close relatives within the same genus that have been cultivated. We have not cultivated them in my lab, but they have been cultivated before. Are these different from those? I don't know. Or do they just, methanogens have been around the Earth for a very long time period. Maybe they just have when it comes to certain temperature conditions, or maybe they have very broad requirements, or but people have cultivated subseafloor methanogens before. There is at least one strain, Methanocoleus submarinus, that was cultivated from the Nankai Trough sediments at, I don't know, 50 meters below seafloor. So it can be done. It requires a lot of patience because you mentioned generation times. They have generation times typically of many months. So that's very difficult for a grad student to do a PhD thesis on something that they will never see in pure culture, or it'll take 20, 30 years maybe.
Yeah, okay, I don't have that much time. Yeah, I usually do E. coli. Yeah, it takes 3 days. Yeah,
Yeah.
okay.
You want fast-growing methanogens, the ones from hydrothermal vents are good for that.
Great toss, and thank you for this talk. It was very interesting. I guess I was thinking about like I think it was on your slide 27. You showed 2 cores. One was like at 43 meters, and the other one is at 37 meters. And you have this steep like shallowing of that sulfate transition zone.
Mm-hmm.
Have you mapped the sulfate transition zone?
Yes.
Across these profiles, and have you seen like a trend with
this is all measured based on measured sulfate concentrations, which are here in black, that black line, and these are measured methane concentrations. So it's based on measurements for each site. Did we map them across the entire geographic region? No, we did not. Some people who were involved in this study have done it for other places, but not for this area. Yeah.
Is that something that you plan to do like in the future? I mean, if you're doing all these cores and like
yes, it would be very interesting. And I've been talking to geochemical modelers, and recently wrote with a proposal with someone in the room here. And so this is certainly this is not what we can produce the data and certain understanding of what's mechanisms, but it would be a great subject of collaboration. Absolutely, and really important for our. I mean, in so many, is a great thing to do.
Thank you.
Thank you. Great talk. So I don't know much about this, but I am interested in the flow. Do you ever think about the connection between the flow velocity, porosity, or can you get sort of connection between the activity of these and the flows?
So these environments we all believe are diffusion dominated. And when there was some initial work by Harry Elderfield on the Juan de Fuca, which suggested that advection through these sediments kind of petered out or became minimal at sediment depths of over 100 meters, if I recall. But you probably know more than I. Here, this is a site with 265 meters of sediment depth. So we generally assume that they're not, and we can actually beautifully model the sulfate concentration profile. So Mark Alpern, a collaborator of mine, has done that for the site, which suggests it's diffusion dominated. And now, what about other? So in these locations, based on my limited understanding, it's not. It's really diffusion that's dominant. But we're looking at other environments on the Azores Plateau, where we think advection is incredibly important, where we have advection of mantle derived and crustal influenced fluids all the way up to the seafloor through actually hundreds of meters of sediments. So I'm not sure why it would work there, not in the Juan de Fuca, but it's very, I think, an understudied and very important project. I mean, research area.
Does the porosity changes across the depth, and is that important or not?
Does the porosity change in these places, or yes, we definitely have compaction, and we also have big changes in grain sizes. So with the Juan de Fuca, we have these turbidites, terrestrial turbidites. We have gravel layers. We're several hundred kilometers off the coast, but we have terrestrial gravel layers, and then we have hemipelagic clay. So we have silt, we have fine sands, we have a wide range of porosities and tortuosities and grain size distributions.
Interesting. Thank you.
Yeah.
Just a quick follow-up to that. And so it didn't look like, and maybe there's a different version where you look at active cells or something, but it didn't look like there was any correlation between that diffusive permeability structure, and all those grain sizes and what life was living there.
Not at all.
That was a surprise to me, at least.
Yeah, it seems like even the lowest, it was the most difficult to detect the organisms in the sandy strata. But you could argue, well, the sandy and gravel strata had the most degraded organic carbon because this is terrestrial organic matter that was part of the mountain ranges and had been through, it's not fresh terrestrial organic matter, but it had been already highly degraded before it became relocated from the terrestrial environment to the deep sea. But those did have it was the most tricky and most difficult to extract DNA, and those strata that were really turbidites were the most difficult to extract DNA from. So the pore size may be great, but if there's no food, then yeah.
The Jets are looking for a quarterback, Sean. So on your last slide, when you say that the lithology doesn't matter, of course, that's something that a geoscientist hates to see. And so I'm going to ask about that. But so in particular, it seems like these guys don't care about any of the kind of the upstream end of things as long as they're getting H+, right? And so I guess my question is, if you were going to go somewhere to look for these beyond like a sedimentary stratigraphic succession off of Peru or someplace like that, has anybody thought about going to places where serpentinization is really active as a place that you might look, like at a slow spreading mid-ocean ridge or a weathering volcanic edifice where you think there's ultramafic rock exposed? Because it seems like that could be a place where you might just sort of shortcut to that source, right?
I mean, if the rocks are producing energy for the microbes, like ultramafic rocks that are releasing hydrogen or methane or acetate or formate, it's a very different story, and that different from these highly weathered mineral grains, which maybe are not releasing significant amounts of microbial electron donors. But there's been work on the Atlantis Massif, quite a bit of work on the North Atlantic and some other region in California. There's some terrestrial serpentinic systems in other places where there's been quite a bit of work on this and suggesting that formate is an important electron donor that's released in these systems. You have chemical reactions between the carbonate and hydrogen. They go towards formate, and then the formate becomes an important electron source for a wide range of microorganisms. They are not typically biomass rich, these environments. Even though they have really high concentrations of formate and hydrogen, that's probably because they tend to be hyperalkaline, oftentimes pH higher than 12, which means it's incredibly challenging for many microorganisms to live there, energetically very, very costly and difficult. So that's what makes those more difficult to live in. In that respect, I think the environment that we're studying in the Azores Plateau might be better. Of course, we're looking at surface sediments, the upper 5 meters. We don't know if that hydrogen or formate that's produced by serpentinization deep below makes it all the way up there. We know the fluids do. We can see the anomalies in magnesium and alkalinity, but whether those electron donors make it up there. But maybe deep down in the sediments, we didn't reach those layers. The conditions are probably buffered better because you have some mixing with the sediment pore water, and perhaps that's where you would have significant stimulation, even maybe deep hotspots of microbial life.
Hey, so in these columns, we can see that there's a variation in the community structure and composition. I was wondering about the community density and if the community, like the amount of biomass with depth is changing at all as you go through these changes in community structure. Some of these communities more prolific or abundant than other communities.
Yes, so that's where when I talked about gene copy numbers of the MCRI genes, we use that as a proxy for biomass. So we're assuming that the cell size, the biomass per cell is the same. Could be that that's not the case. They could vary. But typically when people analyze the carbon content of cells using nanosims, it varies within a small range. Whereas the gene copy numbers go up by 4 orders of magnitude. So clearly, I think if the assumptions are correct, then there has been a strong increase in biomass. And yeah, it's interesting to see which groups show the strongest increases. And one is, again, the methanothrix, the ones that use acetate or potentially direct electrons. Again, the NB1D, which are historically believed to be methane oxidizers, but they seem to always dominate or become abundant when in methanogenesis zones. And then what was a complete surprise is another group called methanomacelicoccalis. These are ones that use methanol and hydrogen as energy substrates. And they were almost absent in the presence of sulfate. But once sulfate was gone, they became very abundant. And again, it's hard to see here, but this is 10 to the 0, that's 10 to the 4, roughly a 4 order of magnitude increase in the gene copy numbers, which we interpret as being evidence of growth.
So I guess kind of a follow-up to that is for the community size or density, what do you believe is the limiting factor? Is it going to be the flow rate of the fluids through those sediments or something else?
So in these diffusion-dominated sediments, I think it's the rate of organic matter decomposition, mineralization. So it's the activity of the fermenters ultimately, and that's driven by the reactivity of the organic matter and the amount of organic matter that's present.
Thank you.
Thanks, really cool. First quick question, I guess I'm familiar with in ice cores, there's a difference in age between the gas and the ice. In sediment cores, you might have a difference in age between sediment and any fluid that's flowing through. In general, when you're thinking about life, what is the age difference between life and the sediment that they're living in?
Great question. We don't know, so the most common, what most people believe, and this is belief, and we can make all kinds of arguments and energetic calculations, and is most of our focus is on diffusion-dominated environments. There's not much flow, and that these environments are so severely energy limited that microbial cells do not have enough energy available to be actually actively moving. So any microbial motion would be by Brownian motion, which is incredibly slow, is negligible. We're talking about millions of years, a few meters maybe. So the reason why, so just to give an example, why energetically it seems unlikely that there's active locomotion is that the microorganisms most microorganisms move by flagellar motions, and flagellum is this protein structure, kind of like a propeller, and it's operates on ATP as its fuel, and for one revolution requires 1 ATP. These organisms are barely getting any. I mean, the amount of, when we do calculations of energy fluxes per cell, there's so little energy. It's based on our calculations. There's barely enough energy to repair DNA damage and replace proteins that are damaged by racemization reactions, and not enough for active locomotion. That would require, according to most estimates, around 1,000 or more times more energy consumption per cell. So that means that they are coming down with the strata. If that's correct, then that's where a lot of people argue that all the microbial cells you find in the deep biosphere were probably deposited with the sediment from above originally, when those layers were deposited. I think that argument is interesting, but I don't necessarily buy it. I think Brownian motion is very slow, but it's fast enough to keep up with sedimentation rates. So I do think you could also potentially have microbial cells entering the sediments, Juan de Fuca ridge flank, from the underlying basalt, and then just keeping up by Brownian motion with the sedimentation rates, which are very, very low over time. So that could be another way that you get over time microbials. I mean, yeah. So I think this is another possibility that has not been explored.
So it could be like 0 years age difference, or it could be as old as the sediments are, like millions of years age difference.
I think it would be very small, because even if it's Brownian motion, it would be only moving a very short distance, and then it'd be the descendants of those cells would be moving. So we're talking about millions of years of sediment accumulation.
Okay. Yeah.
Yeah.
Okay, great. Well, so that connects. I just have one other question that sort of is like a philosophical one, and I'm not super familiar with this. I have colleagues that work on similar things in subglacial sediments, and they're always using these primers to search for like certain genes.
Yeah.
My question, I mean, obviously there's so much that's there, and you have to use the primers. It's helpful to know what you're looking for because otherwise you just have these enormous data sets. On the other hand, you are going in with some kind of either confirmation bias, I'm looking for a certain thing, or, it's possible that over millions of years of descendants, these things are evolving and changing. And so I just wonder from an analytical perspective, how do you balance like going in looking for a particular thing that you've confirmed is important versus that exploratory work that probably defined what you were doing in the early 2000s of trying to figure out there's so much here and we don't have any idea what's driving it. How do we identify sort of the new things that might be important?
I think when we're looking at subseafloor sediments, you have to do both. Ideally, you need to do metagenomics where you sequence everything and combine that with targeted assays, in this case of methanogens, simply because, it could be that there are there are organisms we cannot detect with our primers, but with a shotgun approach, metagenomics, which it also has certain biases, but they're small compared to with primers, you should be able to get them unless they are completely different, have a different genetic code altogether. I will say that in Switzerland, when we did our research on lake sediments, we did both, and we found remarkably good agreement between the MCRA sequences, compositions, and the community structure of methanogens based on metagenomics. Now, whether that's the case elsewhere, I cannot say, but it was a good agreement. And what's also important with primers to always, we have databases that we keep building, and whenever there's new genomes being sequenced, we make sure to import them and make sure our primers are also covering those new genomes. So it's an evolutionary process.
So cool. Thank you.
Mark, that was a really cool talk. And in particular, the fact that methanogens are so rare in deep sea sediments is really surprising to me anyway. And so are there any other data points out there? Like, do we have an idea of the global distribution of these things in deep sea sediments? And if there's a few of them and they're producing all the methane that's being produced, or biogenic methane that's being produced in the sediments, do we have an idea of what the budget is? How much methane are they producing on an annual basis?
So these were the two first community profiles that have ever been published on methane-cycling archaea in subseafloor sediments. Other studies have detected them here and there in ocean drilling cores, but not maybe one. You have 20 samples and you detect them in one layer or two layers. So unfortunately, no. For deep subseafloor, the database is very small, and metagenomics is very difficult. Actually, I'll go back one, because you need a lot of DNA to actually be able to do metagenomics. So a lot of those attempts have not been successful at getting sufficient DNA to really do this kind of exploratory research. Now, with respect to methane production rates, there have been papers by Matthias Egger and Bobacker Jorgensen over the past decade where they modeled based on, it's mostly on methane concentrations in sulfate methane transition zones. What are the total methane production rates in subseafloor sediments? So there's a paper in Nature Communications from a few years ago. That's, to my knowledge, the most up-to-date and comprehensive one.
Do we know if those varied through time?
I would guess they have to. I mean, with primary production being influenced by things like glaciations, right. I think there must be big changes. But to my knowledge, I'm not aware that anyone has done that. It's very difficult to do because you have diffusive mixing of the methane concentration profiles, and methane also, when it's high concentrations down there, you bring it up to the seafloor, you have a lot of ebullition that goes into it bubbles out of the core. So you need to use high pressure, you need pressure coring to maintain the in situ concentrations, and that's dangerous and has only been done in a limited number of places. So that's very difficult to do, but it would be, of course, great to do.
Yeah. Could you improve these records by, like, you found these places where you're able to get enough DNA to do the assay? Could you improve these records by targeting specific regions, or is it just a matter of using enough mud out of the core?
Sorry, I missed that.
Oh, sorry. So to improve the record of methanogens in the deep sea and better understand, how they're producing, is that just, there's like a lot of sediments you just are never going to get good records out of, or if you scraped enough mud out of the core barrel, could you eventually get a usable sample size?
That's a good question. So a big concern with ocean drilling is always contamination. Seawater-based contamination, or if you're using circulating muds doing riser drilling, it's a nightmare. I mean, you have on the Chiku in 2012, when we were off Shimakuita Peninsula, we had 10 to the 9 microbial cells per cubic centimeter of sediment in the drilling mud, which is rich in all these viscosifiers, which are polysaccharides, which are the best food you can possibly give microorganisms. So they're, and we're looking at sediments where there's less than 10 cells per gram. So it's very, very difficult. Of course, with methanogens, it's different because methanogens, well, we think of most methanogens being strict anaerobes, and which you wouldn't find in seawater, and maybe also not in the drilling muds. So maybe you can look at the core barrel. But usually we try to keep our hands away from core barrels, and we stick to sediment cores. We've done contamination tests using microbeads or fluorescent tracers, where we know there's very low drilling fluid intrusion, and we only take the center of those cores. I happen to have a -80 freezer that's full of ocean drilling cores, so we could definitely expand the database with some funding significantly based on those archived cores, and hopefully there'll be more in the future.
Yeah, cool.
Do we have a question online? Okay. Oh, actually, I have 2 questions. The first one is, I'm also thinking about the correlation of the lithology and the methanogens. So do you have any, specific measurements in sand for the cell numbers and the structures of microbial community? So
cell numbers, we assume, and almost all sequenced methanogens so far have 1 copy of MCRA per cell. So it's very convenient. So we can assume, or we assume oftentimes that the number of gene copies corresponds roughly to the number of cells. In terms of their associations with the sediments, no, it's not been studied. It's very difficult to do that. Yeah, it's incredibly difficult actually, because they're so rare. But it's something we have a confocal Raman microscope. We actually want to start looking more at associations between microbial cells and sediment particles, and using isotopic tracer experiments that we can look at labeled cells that are metabolically active, and looking directly at observing directly the degradation of organic matter in sediments. But there's been some attempts, but very little successful work on the deep subseafloor. There's been some work on intertidal sediments where people looked at associations between microbes and sediment grains, but again, it's tricky work to do.
My second question is, what geochemical measurements can we use to understand the in situ activity of microbes?
So oftentimes people measure the concentration gradients of end products and reactants, and then you can model based on the diffusion, based on Fick's laws of diffusion and knowledge on the sedimentary properties. You can model the rates based on that. What many scientists have done is using radioactive tracers. So for instance, 14C labeled CO2 or acetate, or 35S labeled sulfate, and performing incubations in the laboratory where you add these and then perform a short experiment. And after a day or a few days, you measure the conversion of the tracer to a methane or hydrogen sulfide. So there's a discrepancy oftentimes between the rates that are measured based on radio tracers and the ones that are based on modeled geochemical profiles, sometimes orders of magnitude. But yeah, and the question is, why do they not agree? But those are 2 important ones. I also know that Beth Orcutt from Bigelow Labs, they recently developed a new assay to look at broad microbial activity, not specific processes. I forgot how that works actually, but it's not specific to methane production or sulfate reduction.
Let's thank Mark again for the interesting talk and discussion. So if you want to join the lunch with Mark, please meet on the first floor at, I think, 11:40.